Hello everybody, this is the poster presentation containing the results of our project. My name is Greiciane Paneto. I am an associate professor at Federal University of Espírito Santo, Alegre, in Brazil. And these are the initial results of my PhD students, Caroline. And the title of the project is Genetic Differentiation of Citrus using DART sequencing and the development of a rapid species high-resolution melting identification kit. Citrus plants are a diverse group that belongs to Rutaceae family. Among them, the genus Citrus is highly valued due to its economic and nutritional value. However, identifying the species and variety of seedlings acquired by growers can be challenging as the leaves are very similar. To address this issue, we propose using DART sequencing technology to genetically analyze citrus samples and create a rapid species identification kit using high-resolution melting technique. For this, 94 citrus species from the state of Espírito Santo in Brazil were sent to the Service of Genetic Analysis for Agriculture in Saga in Mexico for DARTing sequencing analysis. As a result, a total of more than 69,000 silico-DART and more than 64,000 markers were reported, of which more than 3,000 silico-DART and more than 9,000 SNPs markers were used for analysis after screening with high-quality control parameters. The diversity found was low, but it was as expected. SNPs are being selected using RStudio and BioPython. And here we see a dendrogram showing samples clustering. In blue, we see samples from varieties of sweet oranges, Citrus sinensis, that are most of our samples. And here we see the haplotype network, also showing some clustering. In figure 4, we can see that most of the SNPs are in chromosome 2. No high-resolution melting results yet, because high-resolution melting assay will be developed after SNP selection. This is still in progress. And we can conclude that DART sequencing platforms are indicated as a reliable tool to assign and genomic research on citrus. We are continuing the analysis and the expected results are promising. These are preliminary results. We would like to thank INCAPER, FAPES, CAPES, UFIS for financial support. This is my contact, and thank you very much for your attention.